Hi, welcome once again to our YouTube channel. Thanks for subscribing to our channel. We really appreciate you. And in this particular series, we shall be treating past questions from the Jamb collection. So we'll be looking at a series of questions that have been asked by Jamb over the years, and we'll be giving well-detailed explanations to all of the questions as we come across them. I will implore you to try as much as possible to pay keen attention to all of the explanations that will be given in tackling all of the questions. I wish you success in your exam, and please don't forget to subscribe to our channel. Thank you very much. Hi, welcome once again to our Jamb series. Today we shall be taking questions from the 2021 Jamb biology questions, and so we'll be taking answers to all of the questions as we come across them. So let us see what question 1 is talking about. Question 1 there says, one of the functions of the xylem is A. Strengthening the stem, B. Manufacturing food, C. Reducing loss of water, and D. Conducting manufactured food. Now we're talking about xylem here. So what is a xylem? When we look at the xylem, this is what we'll be coming across. Xylem is a conducting tissue in the plant. Now we have two types of tissues here that we'll be talking about. They are called the vascular bundles. The vascular bundles. And when we talk about them, we have the first one to be the xylem, and its function is to translocate. It translocates water and nutrients from the roots to upper parts of the plant. That is the first vascular bundle, and then the second one that we have there happens to be the phloem. The phloem translocates nutrients and food materials, particularly nutrients that have just been generated in form of starch, from the leaves to the storage sites. So those are the two vascular bundles. Now when we look at these two, particularly the xylem, the moment it absorbs water and it is translocating water from the roots to the upper part of the plant, the cells that are transmitting these xylems become turgid. The turgidity of the xylem gives a strengthening capability to the body of the plant, and that is why we see the function of xylem there to be what we have talked about as option A here, strengthening the stem. So that is the answer to question 1 there, option A. And then we move to question 2. Which of the statements below is not characteristic of wind-pollinated flowers? There are different types of pollination, and when we talk about wind pollination, we're talking about pollination that is carried out by wind. Wind is the agent of pollination in such scenario. Pollination is actually the transfer of pollen grains from the mature anther to the mature stigma. So basically that is what pollination is. So we have different agents of pollination. Agents of pollination now. We have so many of them. We have water, we have wind, we have biological substances like animals, etc. So the particular one we're talking about now is wind. So for wind to be able to pollinate these flowers, or to be able to carry or to transfer these pollen grains from the anther to the stigma, it means the pollens must be feathery, they must be very very light, they must have the ability, they must be very very light, giving the wind the ability to carry them about from place to place. So when we look at the options that we have here, we find out that it says stigmas are usually large and feathery, nectar is usually absent, the pollen grains have rough spiny surfaces, and then the flowers are not scented. Yes, the pollen grains are rough, they have rough spiny surfaces, and that is why it makes it possible for the wind to actually pollinate them, to carry them from place to place. So that is the answer to question 2 there, option C. The pollen grains have rough spiny surfaces. That is option C. So move to question 3. Some of the features of an animal are scales, teeth, nails, and backbone. The animal is likely to be A. Toad, B. Bird, C. Lizard, D. Rat. Which of these organisms here have all of these that we have listed here? Scales, teeth, nails, and backbone. Now, it appears to be that all of them have backbone. The birds do not have teeth, so we cannot pick the birds. The toads do not have nails, so we cannot pick the toads. But which of them now have the scales? Lizard has scales, it has teeth, it has nails, and it has backbone. So that is the most likely fit to this question here. It is going to be the lizard, option C. 3 is option C. And then we move to question 4. Choose the sequence which represents the order of organisms in a food chain. We have grass, snake, toad, grasshopper, hawk. Grass, grasshopper, toad, snake, hawk. Grass, grasshopper, snake, toad, hawk. Grass, snake, grasshopper, toad, hawk. Now, whenever we talk about food chain, we understand that food chain is usually talking about a linear feeding relationship. Food chain is a linear feeding relationship. So oftentimes it must start from the producer and then it moves higher up like that to consumers. We can have primary consumer to secondary consumer and then to tertiary consumer. Now the bottom line is we need to understand that whenever we are looking at these chains, it is usually the transfer of energy that we are looking at. So we are starting with a producer, which is usually the grasses or the green plants, and then what organisms can feed on grasses? It is usually going to be herbivores. So option A here, snake cannot feed on grass, and so option D also, snake cannot feed on grass. So we are taking that option A and option D out. So we are left with option B and option C. So we have grasshopper feeding on grass in the two cases. So what organism will then feed on the grasshopper? It is going to be the toad, which is in option B. We cannot have the snake feeding directly on grasshopper and then toad will now feed on the snake. So it is the toad that feeds on the grasshopper, after which the snake feeds on the toad and then hawk feeds on the snake. So that is going to be option B, which is the most likely correct order in a food chain. Option B, grass to grasshopper to toad, snake, and then hawk. And then we move to question 5. Flatworms and roundworms are said to be invertebrates because A, they are small animals. B, they can live inside the vertebrates. C, some of them are unicellular. B, they have no backbones. Now looking at this, the answer is very very simple. This word that we have here, invertebrate, there simply means without vertebral column. Invertebrate there means without vertebral column. So every other option there which might seem to be the correct answer, they are all wrong. The only one that has a connection with what this question is saying here is the one that talks about the vertebral column, and that is option D. Option D there says They have no backbones. That is the answer to the question, option D. So we move to question 6. When a spirogyra cell is immersed in a salt solution more concentrated than its cell sap, it A. remains unchanged, B. takes tip water and bursts, C. absorbs a little water, and D. loses water and shrivels. Now what we are saying here is that this spirogyra cell is immersed in salt water, meaning that the content of the water it is immersed in is higher in the salt concentration or the salinity than the internal structure of the spirogyra. So when we look at it, this is what we will be having. A cell immersed in, let's do this. If I immerse this cell here in water where we have the surrounding to be highly concentrated, more than what we have inside the cell itself. Now there is a particular phenomenon that will take place here, and it is going to be that this cell here, which has substances inside it but that is of lower concentration compared to the surrounding environment, the substances that makes this environment concentrated will need to be diluted. As such, it will pull water from within the cell. So when water moves from within the cell outside into the surrounding environment, then what happens to the cell itself is that the cell structure starts to reduce in size. We call it shrinking. So it loses water to the surrounding, thereby causing the cell itself to reduce in size, so it shrinks. So that process is known as plasmolysis. So let's see what happens then. They said it remains unchanged. That is not true. It takes some water and bursts. That is not true. It absorbs a little water. That is also not true. It loses water and shrivels. Yes, that is our answer. The cell of that spirogyra will lose water to its surrounding, thereby causing it to shrivel. So that is option D. Question 7. Which of the following is not regarded as a pollutant on land or in the air? A. Smoke, B. Nitrogen, C. Noise, and D. Sulfur dioxide. If we look at this particular question now, we will find out that we most likely might not see any answer here, but there is an answer there. We are looking for a substance that is not actually a pollutant. That is what we are looking for here. We are looking for a substance that is not a pollutant of land and of air. Smoke is actually a pollutant of air. Noise is actually a pollutant of air also. And then we have sulfur dioxide to also be a pollutant of air. The only one that is not a pollutant here is nitrogen. Nitrogen is a constituent of the air that we breathe in. It makes up 76% of the air that we breathe in, or that animals breathe in. So we cannot say that it is a pollutant. Nitrogen is usually found in the environment. Nitrogen is a non-metallic element. It is a colorless, odorless gas that is most plentiful in the elements of the air that we breathe in. So we cannot accept it as a pollutant of the air. So that is the answer to question 7, option B. And then question 8. The initial volume of water poured into a bag of dry soil was 50 ml, and the amount that drained through was 35 ml. The percentage water content of the fully soaked soil is therefore A. 46.7%, B. 25%, C. 20%, and D. 30%. So what is going to be the answer there? They said that the water that was poured into the soil is 50 ml, and then the amount that drained through is 35 ml. So from that we'll be having something that looks like this. This is going to be the working for that question. We have 50%, which was actually poured into the soil, minus 35%, which is the actual water that drained out. So that means the soil has trapped 15 mils of water. They said 50 mils was poured into the soil and then 35 mils drained out. So we are looking for the percentage of water content that is held by the soil. So percentage of water content of the soaked soil is what we are looking for. So we have attested now that 15 mils is what we have left. That is the amount of water that was trapped by the soil. So the percentage of water in the soil now is 15 divided by 50, which is the total amount of water that was poured into the soil, multiplied by 100 over 1, since we are looking for a percentage. So 50 here 1, 50 here 2. So we have 15 times 2, which is going to give us 30%. So 30% is the answer we are looking for. So that is the percentage of the water that is held by the soil. So option D is our answer. And then question 9. When the original king and queen of termites die, they are replaced by A. the king and queen of another colony, B. some adult reproductives from the same colony, C. some adult workers which are specially fed to breed, and D. developing names nurtured as secondary reproductives. Now we're looking at the social insects in this particular question 9. We're looking at the social insects, and we have so many types of social insects, but particularly we have the bees, the termites, and the ants. Now in the termitarium, we usually have the queen and the king, which are usually the reproductives, which bring about multiplication of these colonies. Now in case of the demise of the king and the queen, or the king or the queen, what happens to the termitarium? The termitarium does not cease to exist. What happens is going to be that some of the nymphs, because it is usually a reproductive that develops into the king and the queen. So we find out that some of the names that were nurtured will eventually develop into secondary reproductives, and from those secondary reproductives is where we now have the selection of those that will become the king and the queen. So that is the answer to question 9, option D. And then question 10. In demonstrating the importance of mineral elements in plants, the culture bottle must be darkened to A. prevent algal growth in culture solution, B. allow root growth, C. prevent breakdown of mineral elements, and D. prevent photosynthesis in the root. Now for this particular question here, we find out that we talk about the answer to be the use of this particular bottle called culture bottle to prevent algal growth in culture solution. Now we try as much as possible to use opaque bottles that are not transparent, to prevent the growth of algae in this colony. That is what happens there. We actually use the bottle, that culture bottle, which is darkened. It is not transparent, it is opaque, you cannot see through it. So it is usually used to prevent algal growth, because when there is a penetration of light into the bottle or the content of the culture medium of the culture bottle, there is usually the possibility of formation of growth of algae, and so it will definitely interfere with the mineral experiment that is taking place. So that is the reason why we use amber bottles or culture bottles with darkened color. So it is to prevent algal growth. That is option A. So we'll move to question 11 now. In the mammalian respiratory system, exchange of gases occurs in the A. lungs, B. bronchi, C. bronchioles, and D. the alveoli. We have the exchange of gases taking place in the alveoli. It is in the alveoli that we have the gases, which they are definitely talking about oxygen and carbon dioxide. It is in the alveoli that we have this exchange taking place. Oxygen is taken into the alveoli, and then carbon dioxide is released from the alveoli, and that is how the exchange comes across. So we have that taking place in the alveoli, option D. So question 12. Aged erythrocytes are destroyed in the pancreas, liver, lymph nodes, kidney. We have aged erythrocytes, which are the red blood cells. They have lifespan, and so they usually live for 3 to 4 months, and at the expiration of 4 months, at the expiration of their lifespan, they are actually denatured. And this action is brought about by the function of the liver. The liver and the spleen bring about the destruction of the erythrocytes. So that is the answer to question 12. It is not the pancreas, it is not the lymph node, it is not the kidney, it is the liver. Option B. And then we move to question 13. The villus in the small intestine is significant because it A. increases the surface area for absorption, B. increases the surface area for digestion, C. assists in mixing digested food, D. assists in filtering undigested food. Now, the villus in the small intestine, they are finger-like structures that are found in the small intestine, and their function is to actually help to take nutrients from the small intestine into the bloodstream. Now, why are they very significant? It is because they increase the surface area for absorption. Presence of the villus in the small intestine brings about increase of the surface area for absorption. It is not because it is found in the intestine for digestion, and it does not help in mixing digested food. The answer there is A. It's increasing the surface area for absorption. So that is the answer to question 13, option A. And then question 14. Which of the following food substances will produce a brick red color when warmed with Benedict solution? Benedict solution is a test substance that is used in testing a particular food sample, and the food sample it is used to test is actually carbohydrate. So we have 3 types of carbohydrate. We have the simple sugars, the reducing sugar, and the complex sugar. Now, this particular substance is used to test reducing sugar, and so we can actually use it to test to see if there is a presence of glucose in a food substance. So that is what it is used to test. Now, Benedict solution is actually a blue colored solution, but when it is used to test a substance, depending on the volume or the concentration of this glucose that is in this food substance, if it is a low type of glucose, you find the substance to be changing from the blue color to greenish yellow color. But if the glucose in it is high, if the glucose in it is of high amount, it will change from the blue color of the Benedict solution to a brick red color. So that is the answer to question 14, option A. So we move to question 15. Blood vessels which carries blood from the alimentary canal to the liver is the A. hepatic artery, B. hepatic vein, C. hepatic portal vein, and D. the mesenteric artery. The hepatic artery carries oxygenated blood from the heart to the liver, and hepatic vein carries deoxygenated blood from the liver back to the heart. When we talk about mesenteric artery, it carries oxygenated blood to the intestine. The answer that we are looking for here that carries this type of blood, which is blood from the digestive system to the liver, is the hepatic portal vein. That is the answer to this particular question. The portal vein or the hepatic portal vein, option C. And then 16 there says, which of the following is true of the process of conjugation in paramecium? A. macronucleus disintegrates. B. each X conjugant divides only once. C. macronucleus undergoes division. And D. each macronucleus divides twice. When we look at this question, it's talking about reproduction. And what type of reproduction? It is talking about sexual reproduction. When we talk about reproduction, this is what we'll be having. Reproduction is bringing to life of young ones, and they have 2 types. We have asexual. For asexual, we'll be having these examples like binary fission, we have grafting, we have layering, we have budding, we have another one, let me put it here, we have the vegetative propagation, and vegetative propagation is divided into two. We have the artificial vegetative propagation and the natural vegetative propagation. So those are all asexual. And then for the second one there, we have the for the second one there, we have the sexual reproduction. Talking about the sexual reproduction now, sexual reproduction, basically there are of two types. We have conjugation, which is like what we are having in the question that we are being asked, conjugation. And then the second one is called the fusion of gametes. So conjugation is certainly talking about the process where the conjugates of the cell, substances like the spirogyra or the paramecium, where they actually come to lie side by side with one another in order to carry out reproduction. That is what this particular substance there, that is what it does. So in paramecium, conjugation is formed from sexual reproduction, which is actually a temporary union of two individuals of same species trying to bring about mutual exchange of genetic materials. So the process is actually, like we said, the process is called conjugation. And then two paramecium, they lie side by side, after which they fuse together. They eventually separate and go on their ways in their environment, and then they come to lie side by side again the second time to produce multiple copies of themselves through asexual fission. So that is basically the answer to that question there, that they come to lie side by side. Option D here, they come to lie side by side, each producing micronucleus, and they divide twice. So that is the answer to question 16. Option D. Question 17. People who suck petrol with their mouth run the risk of increasing in their blood the concentration of iron, lead, calcium, magnesium. We have high amount of lead found in the petrol because it is a substance that is used in the cracking of petrol to prevent knocking in engines. So lead is purposefully added to the petrol to crack the petrol in order to prevent it from knocking or to lead the engine into knocking. So that is the answer there. So when these substances, when people suck the petrol with their mouth, some amount of this lead gets into their system, and so they are exposing themselves to lead poisoning. And that is what question 17 is talking about. So in their blood there is going to be the increasing concentration of lead. So that is option B. Question 18 now. Which of the following is a measure for the control of bilharzia? A. Cutting low bushes around homes. B. Application of molluscicides in water bodies. C. Screening windows and doors with mosquito nets. And D. Application of herbicides in water bodies. Bilharzia is actually a pathogen that is actually transmitted by snails. So we actually need to apply molluscicides to water bodies to prevent the infestation of these snails. That is the answer to the question. Option B. And then question 19. The appendicular skeleton is composed of the pectoral girdle and other substances like A. Pelvic girdle, forelimb and hind limbs. Pelvic girdle and forelimbs. Lumbar vertebrae and pelvic girdle. Lumbar vertebrae, fore and hind limbs. Now we talk about the appendicular skeleton. We are actually talking about the skeleton. The skeleton, like we know it, is a rigid framework that gives shape, support, and form to the body of an organism. That is basically what the skeleton is. And we have two types. First one, we have the axial skeleton, which is divided into, we have the brain box or the skull. Brain box or the skull. We have the vertebral column. That is what the axial skeleton is about. And then the second one, which is the appendicular, which is actually what our question is talking about. Appendicular skeleton. Here we have the pelvic girdle, pectoral girdle. We have the forelimbs. We have the hind limbs. The pelvic girdle includes the part that has the acetabulum, which is the femur, the head of the femur, the pelvis, and then the pectoral girdle also have the clavicle. It has the shoulder blade, which is the scapula attached to them. So those are the parts of the appendicular skeleton. So from our question then now, we see that it is already telling us that it has the pectoral girdle. So other part that it possesses or that it has include the pelvic girdle, the forelimb, and the hind limb. So that is option A. And then question 20. A freshwater plant such as water lily can solve the problem of buoyancy by the possession of A. Parenchymal tissues, B. Dissected leaves, C. Thin cell walls of the epidermis, and D. Water repelling epidermis. The question is asking us for the reason why or the possibility of water lily being able to solve the problem of buoyancy. It is simply because of the presence or the possession of a particular tissue called the parenchymal tissue that is actually found in the leaves of the plant. We find such substance in the leaves of the plant, and so it's able to actually encloses air. It traps air in it, such as is made possible for the substance to be able to stay afloat in water or on water. So that is the answer to that question, the parenchymal tissues. Option A. So we move to question 21 now. The correct sequence for the perception of smell in mammals is chemicals, olfactory nerve endings, brain. B. Dissolved chemicals, nasal sensory cells, brain. C. Chemicals, mucous membrane sensory cells, brain. D. Dissolved chemicals, sensory cells, olfactory nerve, and then the brain. When smell is to be perceived, there is going to be the need of the substance to be perceived. So it actually starts from that substance. So we have dissolved chemical, and then it moves to the receptors of this smell. The receptors of the smell are the substances that actually receive this stimuli, and they happen to be the sensory cells. The sensory cells around the nose help to receive this stimuli. So the dissolved chemicals are received by the sensory cells, and then they are sent to the brain through the olfactory nerves. They are sent to the part of the brain called the olfactory lobe through the olfactory nerves. So that is the answer or the sequence of smell perception. Option D. It moves from the dissolved chemicals to the sensory cells and then to the olfactory nerve endings to the brain. Option D for question 21. And then question 22. Which combination of these can cause rapid overcrowding in climax biotic communities and human settlement? 1. High birth rate and high immigration rate. Roman numeral 2. Low birth rate and high immigration rate. Roman numeral 3. Low birth rate and low immigration rate. Roman numeral 4. High mortality rate and high immigration rate. I will quickly put the answer here. Let me not go too far to make it easy for us. If you look at the first one there, says high birth rate. So number 1 there, when there is high birth rate, it is definitely going to increase the population of that community. And then high immigration. When there is high birth rate, there is increase in the birth record natality of that environment, so it increases population. And then high immigration means people are coming into that community, so it will also increase population. That is the possibility of one. Two there says low birth rate. Low birth rate is going to reduce population, so we have it reducing population. And then high immigration, so this is possibility of what we have for the second one. So for the third one there, low mortality rate means that there is decrease in death rate and then low immigration. So decrease in death rate will definitely increase population. And then low immigration means the number of people that are going out will reduce. So even if it doesn't reduce, it is similar, it is going to be somewhat low. And then the last one that says high mortality rate, that is there is high possibility of people dying, so that is option 4. And then high emigration. So the only one that can actually increase the population, like we have seen in our question there, which is talking about possibility of the community having what we refer to as overcrowding in the climax biotic community, is when we have number one, Roman numeral 1 is the only option that is increasing in both scenarios. So high birth rate and high immigration rate. And then we have the third one there also, low mortality rate and low immigration. When mortality is low, we find it that it is going to result to increase or stability in the possibility of that community. And then low immigration also will result to normalcy or increase in the population. So our options there, we have Roman numeral 1 and Roman numeral 3. This one here is supposed to be increase also. This one here is supposed to be increase. So we have it to be increase in both scenarios. So this one is correct, and then this one also is correct. So Roman numeral 1 and Roman numeral 3. So those are the possibilities. That is option B. And then question 23. The number of plant species obtained from a population study of a garden is as follows: guinea grass 15, hypomoea species 5, cedar species 7, and imperator species 23. What is the percentage of occurrence of imperator species? We want to know the percentage of occurrence of imperator species in all of these population sample or study that we have taken. So let us quickly work that out. The first one, we have guinea grass to be 15, hypomoea species 5, cedar species 7, and then imperator species to be 23. So let's quickly have their total: 15 plus 5 plus 7 plus 23. That will give us a total of 15 plus 5 is 20, 7 plus 23 is 30. So 20 plus 30 will give us 50. So 50. So we are looking for the percentage from the question there. It is asking us for the percentage of occurrence of imperator species, and this is for imperator species here. We have imperator species to be 23. So that is what we are looking for in that question there. So 23 over 50 multiplied by 100 over 1. So 50 here 1, 50 here 2. So 23 times 50 is going to give us 46%. So that is the possibility or the percentage occurrence of imperator species, 46%, which is option C. So question 24. When a marine fish was taken from the ocean and put in a tank of fresh water, it died after a short period because A. The tank was too cold compared to the large ocean. B. The body cells of the fish swelled and burst as a result of the hypotonic fresh water. C. The body cells of the fish shrank as their sap was hypertonic to the fresh water. And D. There was no food in a tank, so the fish starved. Remember that marine habitat is an habitat that is full of salt water. The salinity of salt water is very, very high. So the fish that is taken from that environment into the water environment, is going to be moving from, naturally the environment of that fish was originally high. It had hypertonic solution. The fish was in an hypertonic solution, and then it is taken out and put into fresh water, which is going to be hypotonic solution. So what happens in such like this one here? So when we have the substance here and the fish is dropped into this environment here, we notice that the fish here is going to, since it was dropped in environment where it is of lower concentration compared to where it was coming from, now the substances here inside the fish will need to be neutralized, or will need to reduce its concentration. So it is going to absorb water to the body of the fish. So water moves into the fish from the surrounding. As such, there is going to be what we refer to as the bloating of the fish, where the fish will absorb so much water until the fish starts to burst. So that is the answer to that question there. It says that the body cells of the fish swelled and burst as a result of the hypotonic fresh water. So that is the answer to question 24, option B. So question 25. If a DNA strand has a base sequence of TCA, its complementary strand must be A. ATG, B. TAG, C. GAT, and D. AGT. Okay, let me just put it here. DNA, the complementary strands that we have it looking like this: A, G, T, C, A, T, G, A. Now for all of this substance here, every time we have A, the complementary strand is going to be T, and every time we have G, the complementary strand is going to be C. So T to A, C to G, A to T, T to A, G to C, A to T. So that is the complementary strand that we have. That is how we have them. So from our question now, it is asking us that the complementary strand for TCA is going to be what? T. The complementary strand of T is going to be A, and then for C it is going to be G, and then for A it is going to be T. So we have it to be AGT. AGT, that is option D. And then question 26. The two key cations involved in the action potential of nervous transmissions are sodium ion and ferrous. We have magnesium and potassium. We have sodium and potassium. We have iron 2 plus and magnesium 2 plus. Now the substances that we have there that are responsible for potential difference or action potential across the nervous transmission is the sodium potassium, and that is where we have what is referred to as the sodium potassium ATPase. Now if I were to do this, I would draw something like this. Imagine that we have a body like this, and then we have the body divided into two here. Now all this place here, we have sodium, and here we have potassium. So for this to come out now, this is inside the body. Let me put it like this. Inside the cell, and here we have outside the cell. So for this substance that is inside the cell to get out of the body, there is going to be that is where we have this substance that is called the sodium potassium ATPase. We call it sodium potassium ATPase. So there's going to be the need for this substance here called the wheel to spin. So every time that there is a spinning of this wheel here, we have something like this. So when this wheel spins like this, we have this substance here, the sodium that is coming in pushing out the potassium that is inside the body to the outside. So that is what brings about that substance there, and so it is what brings about the action potential. So it is sodium potassium. That is option C. And then question 27. The order of evolutionary advancement of the vertebrates below is Ratus ratus, Agama agama, Bufo regularis, Tilapia zele. Now when we look at this particular answer here, this particular question here, we find out that the answer is going to be option D, which is going to be the Tilapia zele, the Bufo regularis, the Agama agama, and the Ratus ratus. Why? Because the evolutionary trend of organisms, particularly vertebrates, we have it like this. The Pisces to the amphibians, and from the amphibians to the reptiles, and from the reptiles to the birds, and then from the birds to mammals. So from the question there, we have Ratus ratus, which is rat. We have Agama agama, which is the agama lizard. We have the Bufo regularis, which is the toad, and then we have Tilapia zele, which is the fish. So here, rat is a mammal. We have the fish here. We have the toad here. That is the Bufo regularis, and then Agama agama, which is the agama lizard. So if you look at it now, we have fish to toad to agama to rat. So that is the answer to that question. Fish to toad to agama to rat. So we see it now is the fish to toad to agama and then to rat. So that is option D. 4, 3, 2, 1. Option D. And then 28. Which of the following produces both hormones and enzymes? A. Ileum, B. Pancreas, C. Gallbladder, and D. Kidney. It is actually the pancreas that produces hormones, and at the same time it produces enzymes. The pancreas produces digestive enzymes, and then the digestive enzymes we have the amylases, we have the tripases or trypsin, and then we have the lipases. Those are the three digestive enzymes that it produces, and then it produces hormones as well, because it is an endocrine gland. So the hormones that it produces insulin, which is responsible for converting excess glucose in the blood into glycogen. It produces insulin, and so the glycogen is stored in the liver, and when the body needs this sugar again, the pancreas produces another hormone again, which is called the glucagon. So that is why we call the pancreas a hormonal gland, as well as it producing digestive enzymes. So that's the answer to that question there, the pancreas, option B. And then question 29. The correct sequence of movement of urea during urination is A. Glomerulus, Bowman's capsule, convoluted tubule, Henle's loop, collecting tubule. B. Glomerulus, Bowman's capsule, convoluted tubule, Henle's loop, convoluted tubule, collecting tube. C. convoluted tubule, glomerulus, Nettles loop or Ellis loop, Bowman's capsule, collecting tubule, and D, convoluted tubule, Bowman's capsule, Ellis loop, glomerulus, and then collecting tubule. The answer there is this one here, which is glomerulus to Bowman's capsule to convoluted tubule, Henle's loop, and then the collecting tubule. That is the system of urine formation. You know, when we talk about urine formation, basically we have three processes, and the three processes that we have in the process of urine formation, we have the glomerular filtration, and then we have reabsorption, as well as the tubular secretion. The kidney tries as much as possible to prevent water loss to the surrounding, particularly when the body is dehydrating. It tries to prevent water loss, excess water loss to the surrounding, and so it carries out selective reabsorption. It also does not want to lose major materials to the body, and so it filters unwanted substances, takes them out of the body in urine, while needed substances are taken back into the body in the process of reabsorption. So that is basically the answer to that question 29, option A. And then question 30. The order of passage of food in the digestive system is ileum, cecum, large intestine, rectum. Ileum, colon, cecum, rectum. Large intestine, ileum, cecum, rectum. And D there says colon, cecum, ileum, rectum. Now, the ileum is the small intestine. From the small intestine, we have the food passing through into the large intestine through a particular passageway called the cecum, after which the large intestine sends out the waste materials in the body in form of pellets as sent out in the rectum. So that is the answer to that question there. The food passes from the ileum into the cecum, the large intestine, and then into the rectum. Option A. So question 31. Mass of crucible equals 10 grams. Mass of a crucible and soil before heating equals 29 grams. Mass of crucible and soil after heating equals 18 grams. From the information above, determine the percentage of water in the given soil sample. Now, the mass of the crucible alone is 10 grams. Then the mass of crucible plus the soil before heating is 29 grams. And the mass of crucible after heating has taken place is 18 grams. Mass of crucible, we have it to be 10 grams. Crucible plus soil before heating, 29 grams. Then crucible plus soil after heating, we have it to be 18 grams. So that means here we have the soil before heating. Soil before heating equals 29 minus 10. 29 minus 10 grams, so that gave us 19 grams. And then soil after heating. Soil after heating equals, what we have here is the soil after heating, 18 minus weight of the crucible, which is giving us 8 grams. So the difference between the soil before heating and after heating is the removal of water. So water has been removed in the process of heating the soil. Evaporation took place, so we have the difference to be the amount of water. So amount of water vapor equals 19 grams minus 18 grams, so we have it to be 11 grams supposedly. So the question now is asking us, it said determine the percentage of water in the given soil sample. The percentage of water in the given soil sample. So we have the percentage of water to be, we have it to be 11 over 19 times 100 over 1. 11 over 19. Why are we picking 11? 11 is the amount of water that we got, and then 19 is the total weight of the soil plus the water. The weight of the soil plus the water is what we have here as 19 grams. So we have 11 over 19 multiplied by 100 over 1, which is our percentage. So when we carry out our calculation, we will discover that 19 from 100 is going to give us 5 points, and then 11 times 5.34, so it's going to give us 60%. So that is going to be the answer to that question there. So we have it to be 60%, option D. So question 30. Which of the structures is correctly matched with the organisms in which it is found? Stomata spirogyra, alveoli earthworm, malpighian tubule mammal, contractile vacuole, protozoan. Stomata is seen in plants. It helps to actually carry out the exchange of carbon dioxide. It allows plants to take carbon dioxide in, and it is actually needed for photosynthesis to take place. Also, the stomata uses water loss. So that is the function of the stomata. So it is not found in spirogyra, it is found in the plants. And then alveoli and earthworm. We don't see alveoli in earthworm. The alveoli are found where there are lungs, and the blood exchange of oxygen and carbon dioxide takes place in the alveoli during the process of breathing in and out. And then we have the third one there to be malpighian tubule and mammal. Mammals don't have malpighian tubule. Malpighian tubule is the excretory organelle for insects, so it cannot be found in mammals. And then the last one there, we have the contractile vacuole for protozoans. Yes, contractile vacuole is actually the excretory organelle for the protozoans, for the unicellular organisms. So that is the answer to our question there, Roman numeral 4, which is option C. And then move to question 33. The sequence of the one-way gaseous exchange mechanism in a fish is mouth, operculum, gills, mouth, gills, operculum, operculum, gills, mouth, gills, operculum, mouth. The oxygen that these organisms here, talking about marine organisms, particularly the fishes, the pisces, the oxygen they breathe in is in dissolved form in water. So they actually take in water to get their oxygen. So how do they now carry out the extraction of the oxygen from the water? The water goes into their mouth. The water goes into their mouth, and by the time the water is transferred to the gills use its system to actually carry out the extraction of the oxygen in the dissolved form in water, and then it expels the remnant of the water through the operculum in the sides of the body of the organism, talking about the fishes. So the system is actually through the mouth to the gills and then the operculum. Option B is the answer to question 33. And then we have question 34. Which of the following is true in blood transfusion? A person of blood group AB can donate blood only to another person of blood group AB. B, persons of blood groups A and B can donate or receive blood from each other. C, a person of blood group AB can receive blood only from persons of blood group A or B. And D, a person of blood group O can donate only to a person of blood group O. Looking at this now, we need to understand the possibility of blood donation in organisms. So I'll put that here. We have basically 4 blood types: A, B, AB, and O. So when we look at the characteristics of these four blood types, we will see that they look like this. So talking about their characteristics now, we have the antigens, in the RBC, and we have antibodies in the WBC. Now for A has antigen A and it has anti-B antibody. That means it is against B. So any blood type that has antigen B will not be able to donate blood to A. B on the other hand has antigen B, and so it has anti-A antibody. It has antigen B, but it does not receive blood from blood types that have antigen A. That is the meaning of this anti-A here. And then AB blood type here. AB blood type has antigen A and antigen B. It has antigen A and antigen B, but it does not have any antibody. So it is negative for the antibody. And then that means since it does not have any antibody, it can receive blood. It is not against A and it is at the same time not against B. So it can receive blood from any blood type that has antigen A or antigen B. While O here, O does not have any antigen. O does not have any antigen, but it has anti-A and anti-B antibodies. It has anti-A and anti-B antibodies. And that is why when we look at the donation pattern, when A is to donate blood, this is how the donation looks. Let me put it here. A, B, O. And then we now have A, B, AB, and O. So this one here happens to be the recipients, while these ones here, they are the donors. So A can donate to A because A has antigen A, and that is what A needs. So it can donate to A. It does not have the A that is donating has antigen A, and that the A that is receiving is against B, not against A. So it can actually take blood from it. And I have the second one there, which is actually B. A, which has antigen A, is donating to B, which has anti-A antibody. So since B has anti-A antibody, it cannot receive blood from a blood type that has antigen A. So this one will not work. And then A can donate to AB because A has antigen A, and AB that is receiving does not have any antibody. So this one will work as well. A that has antigen A here cannot donate to O because O here is against A. You can see the chart here against A. So it means this one will also not work. And then B can donate to B. But let's take A first. B cannot donate to A because B here has antigen B, which is what the A has as antibody. A has anti-B antibody. That means it is against B. That is the antigen that B is coming with. So this one will not work. But B can donate to B, and B can donate to AB like A was able to donate to AB. But B also cannot donate to O because B has antigen B, while O has anti-B antibody, so this is not possible. AB cannot donate to A because even if AB has antigen A, it also has antigen B, and because the blood type A, which is the recipient, has anti-B antibody, it will clot with the B factor coming from the AB blood type, so this is not possible. And similar fate is what happens to AB donating blood to B blood type, but AB can donate blood to AB, it will not also be able to donate to O. And then for O can donate to A since O does not have any antigen, so it can donate to A will receive that blood from O, and then O can also donate to B since O does not have the antigen as well. It can also donate to AB and it can also donate to O. So we see AB, we see them as universal recipients. If you look at it now, you notice that everything here is what we can match on. So that is the universal recipient. While the second one there, which is O is seen as, we call them the universal donor. You can see O here. All of the tracks here, they are all checked. So that is why it is called the universal donor. So O can actually donate to all the 4 blood types. So looking at this question here now, it is asking us that which one is true. A person of blood group AB can only donate to another person that has blood group AB. AB cannot donate to any other blood type. So A is absolutely correct. Persons of blood group A and B can donate or receive blood from each other. No. A here has antigen A and it has anti-B antibody, and B here is having antigen B and anti-A antibody. These two substances, these two blood groups here cannot donate blood to themselves because they have antagonistic antibodies. And then option C there says a person of blood group AB can receive blood from persons, only from persons of blood group A or B. No, the word only here nullifies this statement here. AB cannot receive blood only from A and B, or A or B. It can receive blood even from blood group AB as well as blood group O. So this statement here also is not correct. And the last one there says a person of blood group O can donate only to a person of blood group O. This is also not correct. A person of blood group O can donate to every other blood type. So the only one that is correct in this statement here is the blood group AB, which can donate to another person of blood group AB. That is the option there for question 34. And then question 35. The plants that grow in deserts or very dry areas are referred to as A. Hydrophytes, B. Epiphytes, C. Xerophytes, and D. Mesophytes. Hydrophytes are plants that grow in environments with abundance of water. So they are oftentimes called water loving. Epiphytes grow on other organisms. We have the mesophytes, which are plants that grow in areas with moderate moisture that is not too dry nor wet. And then the last one is called the xerophyte. They are organisms that survive in environments with extreme temperature and low water content. So that is the answer to our question there. Substances that survive in desert environment, places that have dry surroundings, they are called the xerophytes, option C. And then question 36. The most abundant group of organisms in animal kingdom: A. Mammal, B. Apes, C. Insecta, D. The annelidas. It is the insectas. The insectas have more organisms in their class or in their group. The answer there is the insecta. It is not the mammalia, it is not the apes, it is not annelida, it is the insectas. Option C. And then we have question 37. The movement of parts of a plant in response to external stimulus of no particular direction is taxism, tropism, haptotropic movement, nastic movement. When we talk about this, we're talking about responses. And we have basically three types of response. Types of response. Number one, we have the tropic or tropism. This is carried out by plants. Plants response to directional movements. And then the second one, taxic or taxism. This is animal response to directional as well. Directional movement. And then we have the third one to be nastic or nastism. This one is movement of the both of them, both responding. When I say both now, I mean plants and animals responding to non-directional movements. So that is what these three are. So looking at the question now, the question is saying the movement of part of a plant in a response to external stimuli of no particular direction. Since it is no particular, so it means it is non-directional. So that tells us that it is a nastic movement, not tropism. If it were to be a directional movement, it would have been tropism, but since it is of no particular direction, it is a non-directional movement, so it means it is the nastic movement. That is option. And then question 38. The mutation theory of organic evolution was propounded by Gregor Mendel, Hugo de Vries, John Lamarck, and Charles Darwin. Gregor Mendel was the one that actually talked about genetics. Gregor Mendel was the one that actually started the study of genes. And then when we talk about John Lamarck actually talked about the transfer of acquired traits from parents to offspring. That was the contribution of John Lamarck to the evolution theory. And then we have the last one, which is Charles Darwin. Charles Darwin was responsible for talking about heterozygous and homozygous traits. Charles Darwin actually helped to throw more light on what the origin of species, what it actually is. But the person that actually talked about the mutation theory of organic evolution is Hugo de Vries. That is the person that actually made that contribution. So that is option B. And then question 39. If both parents are heterozygous for a trait, the probability that an offspring will be recessive for that trait is half, quarter, 1, 3/4. Let us see. Both parents are heterozygous for that trait. So let us see the possibility of that happening for the father and this for the mother. So when meiosis takes place, it breaks up, and then there's going to be recombination of gene such that we'll be having something that looks like this, where we'll be having HH. We will also be having this. We'll also be having this. And lastly, we'll be having this. So the question now is, so these are the possibilities that we will be having. We'll be having 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. So the question now is, the question said, the probability that an offspring will be recessive for that trait is, since we have that particular substance, if it is going to be recessive, this is what it is going to be looking like. This one's here, we have homozygous dominant. This one's here that heterozygous, but it is still dominant. So this is the only one that is actually showing the recessiveness. This one here. This is the only one that is showing the recessiveness. So it is one of 4 possibilities. So 1 of 4 possibilities. So that is what we are having. So that is the answer to that question. Option B, that is quarter. And then we have 40, which is the last question there. 40 there says, blood circulation in a mammal is said to be double because A, it passes twice through the heart in complete circuit. B, it moves in both arteries and veins. C, it circulates in both the heart and other organs. And D, the heart contains auricles and ventricles. Why? Because the blood passes the heart twice. First and foremost, we need to understand that there are 3 types of hearts. 3 types of heart. The first one, we have the 2-chambered heart. This one here has 1 auricle and 1 ventricle. Some textbooks will call this auricle, they call them atrium. Now, the second one there, it has 3-chambered heart. So it has 1 auricle and 2 ventricles. And then the third one there, we have the 4-chambered heart, where we have the 2 auricles and 2 ventricles. For the 2-chambered heart, example of organisms that have 2-chambered hearts, we have the fishes. Fishes have 2-chambered hearts, and then for the 3-chambered hearts, we have the amphibians. And then for the 4-chambered heart is where we have the mammals. So it is in this four-chambered heart now that we now have what we refer to as the pulmonary and the systemic type of circulation. So that is what I'm putting down here now. We have the heart here, and then we have the lungs here, and we have other organs here like the kidney, the liver, the intestine, and so many other organs that we can think of here. So we are having blood coming from the heart to organs of the body, and coming from the heart to the lungs. Now when we look at it here now, this particular one that is taking blood away, all vessels taking blood away from the heart, they are called arteries. So all these ones here, they are arteries. This one too is taking blood to the heart, so it is called, taking blood to away from the heart rather. Vessels that take blood away from the heart to organs of the body, they are called arteries. And the ones that take blood back to the heart, they are called veins. So we have this now. Now, these ones here that take blood from the heart to organs of the body, they are carrying oxygenated blood. They are carrying oxygenated blood. And then the one that is taking the blood from the heart to the lungs alone is carrying deoxygenated blood. And then on the other side here, the vessels that are taking blood from the organs of the body back to the heart, they are carrying deoxygenated blood. And then the one that is carrying blood from the lungs back to the heart is carrying oxygenated blood. So when we look at it, every artery of the body, excluding the one that is carrying blood to the lungs, this one here, this particular artery here, every other artery that is supplying the liver, the kidney, the intestine, every other artery supplying other body organs, excluding the one that is supplying the lungs, they are all carrying oxygenated blood. The only one that is not carrying oxygenated blood is the one that is supplying the lungs. That one carries deoxygenated blood. So that particular artery is called, this one here is called pulmonary artery. The one that supplies the lungs is called pulmonary artery. Now every other vein also that is taking blood back to the heart, every vein that is taking blood back to the heart, excluding the one that is coming from the lungs, they are all called the veins. But the one they all carry deoxygenated blood. The only one that is taking blood from the lungs to the heart is called the pulmonary vein, and it is the only vein that carries oxygenated blood. Pulmonary vein. So this is what I'm talking about here. It carries oxygenated blood. So pulmonary vein is the only vein that carries oxygenated blood, and it comes from the lungs to the heart, while the pulmonary artery is the only artery that carries deoxygenated blood, and it comes from the heart to the lungs. So now our question is, why do we say that the blood passes the heart twice? Or the question that we're actually talking about here, that the circulation of blood in mama is said to be double. Now it is because whenever we breathe, oxygen is being taken in the lungs into the blood. The lungs mixes when the blood the lungs, the alveoli of the lungs carries out oxygen or gaseous exchange. It removes the carbon dioxide in the blood and it replaces it with oxygen, and then the lung sends that oxygenated blood through the pulmonary veins. Now it sends the oxygenated blood through the pulmonary veins to the heart. When the heart gets the blood, the heart now pumps that oxygenated blood to organs of the body through other arteries, and so that is why other arteries are carrying oxygenated blood. Now when these arteries take the blood to organs of the body, these organs of the body, they have previously used some gases before, so they have some waste materials, particularly gaseous waste material, which is definitely going to be carbon dioxide. So they take the oxygen that is in the blood that is coming from the arteries, they remove the oxygen and then they replace it with carbon dioxide, and then these veins here, they take that deoxygenated blood now back to the heart, and when the blood gets back to the heart sends it to the lungs, so that the lungs can actually remove the carbon dioxide and replace it with oxygen. So the first time the blood comes into the heart is when it comes from the lungs. When it comes from the lungs into the heart, that is the first one, and then it goes like this into the heart pumps that oxygenated blood into organs of the body, and then the blood goes back to the heart the second time. Now when the carbon dioxide has been put in the blood, it goes to the heart and then the heart takes it back to the lungs for expulsion. So those are the two times that the blood gets to the heart, and that is why it is called double circulation. So the blood passes twice through the heart in a complete circle. That is the answer to 40th question there, option A. So that brings us to the end of this particular question series. I'll implore you to try as much as possible to pay keen attention, watch this video once more or as many times as possible to be able to get to understand all that has been discussed. To those of us that are yet to subscribe to our channel, I'll implore you to please do so, because you'll be gaining tremendously from subscribing to the channel. And don't forget to click on the notification button so you get easily notified when we upload new content. Like our content, share them with friends, let them benefit from what you have also gotten. Also, we want to hear from you, so we want your questions in the comment section. Don't forget to send them across to you and across to us, and we promise to get back to you as soon as we can on all the questions that you send to us. I wish you success in your exams and best of luck. Bye for now.